Okay, so again, I just repeat that. So in this lecture, we're actually dealing with diversity and immunological tolerance, which are actually crucial aspects of the adaptive immunity. So because our body constantly encounters different antigens every day, all right? And in order to do so, our body needs to recognize them. And how to recognize them is to generate them from your genes. And in doing so, sometimes this opens up to another problem, is that some of these T cell receptors, MHC molecules, or antibodies, they actually can bind to our own antigens. And when that binds to our own antigens, this actually will elicit, the immune responses of cell-mediated immunity or humoral immune response. And this actually will cause autoimmunity. And to prevent that from happening, our body actually has this thing called immunological tolerance. So why is it important? Because to prevent autoimmunity. And why? You'll see afterwards. The reason we have immunological tolerance. And what is central tolerance and peripheral tolerance? What are the mechanisms, okay, inside that? And this VDJ recombination, so this is actually part of the gene rearrangement, and discuss the consequence of loss of tolerance. So first, we need to define immunological tolerance, which means unresponsiveness to an antigen that is induced by previous exposure to that antigen. And antigens that induce immune response are known as immunogens, vice versa for tolerogens, whereby they are antigens that induce tolerance. So, in the previous lecture, we covered this. Lymphocyte plus antigen activated, leading to immune response. However, for immunological tolerance, you see this. Either it gets inactivated or eliminated or deleted. And you also got special cases. So, failure of self-tolerance leads to immune response to self-antigens. And self-antigens, so this self here, some people have different names, actually called autologous antigens. And failure of that leads to autoimmune disease. So it's a physiological state where the immune system does not react destructively against the host. It involves many interactions between immune cells and other cells in a well-regulated and controlled manner. You'll see how. And allows discrimination between self and non-self antigens. So no friendly fire allowed, okay? Only against non-self and avoid against self. So the reason for immunological tolerance, yeah, sorry about this. It's actually quite wordy here. So in order to cover a quite diverse repertoire of different antigens, so receptor needs to be as large as possible. But however, in doing so, through this VDJ recombination process, random shuffling of many genes, okay, to encode this actually allows the generation of all these 10 to the power 16 different combinations. So power 6 is million, another 12 is billion, right? Then add another 4, so another 1,000 billion. That will be 1 trillion, right? Roughly. So again, about 1 trillion different combinations. However, just like, you always have errors. And some of these combinations will target self-antigens. And to do so, we have central tolerance and peripheral tolerance. So central tolerance, as the name implies, occurs at the generative lymphoid organs of the thymus and the bone marrow. Peripheral tolerance occurs in the peripheral circulation, normally. And generally, this is general. These are the mechanisms for immunological tolerance. Got deletion, energy, suppression, receptor editing. So these are the general ones. Okay, but the emphasis is here, these two, and the special cases for T cells and B cells respectively. So this is just a figure showing you our central torrents and peripheral torrents. So as you can see, apoptosis energy. Actually, an energy can also occur here as well. Apoptosis also occur, as you can see, apoptosis can also occur in peripheral torrents. Occurs in both. Then for B cells, you have receptor editing here, and here suppression is your T regulatory cells. Even though T regulatory cells are actually developed here, they only do their work in peripheral torrents. Okay, so they are actually part of the peripheral torrents here, your Tregs. Okay, there are actually many factors that can affect torrents. Okay, so this is actually a very good table. I actually found this. I think it's actually very good, even though some of the terms here you probably haven't encountered before. Okay, what's most important here is this one, this one. Altogether, you can group them as environmental. This one at the bottom here is actually genetics or internal factor. Okay, so example, hormonal balance, stress, drugs. So what happens? So hormones, some actually hormones like estrogen, they can actually cause problems. They can actually suppress the immune system. Stress, if you still remember your cortisol. So stress actually can downregulate the immune response, and so is certain drugs. Okay, so fresh flows, what does it mean? Altered antigens. So some antigens, they are changed. So as you can see, attachment by hapten to make a new antigen. Or they can actually mimic. So the foreign antigen, they mimic self-antigen. Unmasking. So exposure of previously sequestered self-molecules. Anyhow, all in all, always just remember, environment factors can affect torrents, and so do we have internal factors. Okay, so this is actually just for your reference for now. Okay, so now we're actually going to go to into detail of the different central torrents processes for T cells. Okay, happens in the thymus, even though they originate from the bone marrow, but they still go through central torrents in the thymus. Now, as you can see here, from the bone marrow, start off as DN, double negative cells. So see here, CD4 negative, CD8 negative, T cell receptor negative. What does this mean? Means at this point, they still haven't have the receptors, the cell surface markers, okay, necessary for them to be called T cells. So they just need to go to the thymus. So imagine this as a high school. Now they go to the university, and here in the university, they need to go through a bunch of selection processes. So these selection processes is like your exam, okay, just that, very stringent. Failure of that exam leads to death. Okay, unlike med school you resit. So a bit different. So, and during this selection process, they actually undergo a lot of this thing called genetic rearrangement. The VDJ recombination happens here as well. And all in all, the idea between all this is to generate this, an SP, a single positive cell. So what does it mean by a single positive? A single positive means single is either CD4 or CD8 T cell. And this one is the basic. You need to have this all the time, because I'm sure by now you know the importance of T cell receptor as well as CD3 positive. So again, so during in the thymus, during the development of the T cells in the thymus, involve many intermediate stages. I won't go into detail with this one. Very complex. Just that you have this genetic rearrangement as well as a VDJ recombination. Okay. leading to expression of a functional T cell receptor. Then they need to go through two selection processes, excuse me, positive and negative selection. So this is your exam, okay? And now ultimately, commitment to phenotypically and functionally distinct subsets, a CD4 positive or CD8. So either way, you end up with a T helper cell or a cytotoxic T cell.
So doctor, if we know just this for the CA right now, it's sufficient, or do we know it? Need to know it in detail? The development of.
Well, we haven't gone through this yet.
Oh, okay.
So for my part, just focus on what's here. I won't bother with this one. I actually bother with the importance of why, during the T cell development, why this happens. Why? I prefer that. I won't want you to remember details like pre, pro, or what. But never mind. Let's continue. Okay, some terms you might feel a bit, if you still feel a bit lost. DP, double positive. SP, single positive. DN, double negative. So, as you can see here, everything starts off as a double negative stage. So what does it mean by double negative? Is CD4 negative, CD8 negative. And here, what happens during this stage, the genes are still in germline configuration. So they're still there waiting, okay, before any rearrangement process. Because after this, you'll see the gene rearrangement. So double negative T cells refers to cells without T cell receptor and CD4 or CD8. So this is how it looks like, the T cell receptors in germline configuration. Still remember, you got all these different segments, okay? 70 to 80, J times 61. Then you got alpha chain, beta chain. What does this mean, alpha chain, beta chain? Still remember your T cell receptor. You got alpha, beta. This you got another one, gamma, delta. We focus on this one. So as you can see, you got all different types of all these potential combinations. And in total, you got roughly 1 trillion of them. So this is actually before rearrangement occurs. Okay, so I'm actually going to show you how rearrangement occurs. So as you can see, this is your alpha and this is your beta chain. And this process is mediated by this thing called recombination activating protein. Okay, which results in formation of the T cell receptor. This is actually quite simple already. Okay, I don't want to go through in detail on this one. But again, crucial process here is this one. Important enzyme. Then all I really want you to appreciate here is also that this will result in total diversity here. So this is actually just the potential, calculation. By now, still don't go into this one lah. So as you can see, this is antibodies, and this is the T cell receptor. So see, you got 1 trillion for both. Then you can see here variable diversity joining, because here as you can see variable joining constant diversity. So this is what it means, the VDJ, all that. So it is just for your reference. Now, during gene rearrangement, everything is random. Simply do. Okay? Why? To increase diversity. However, in doing so, this actually causes a lot of problems down the line. And as you can see later, so double positive cells, CD8 positive, CD4 positive T cells. Same can be observed in B cells, particularly for the B cell receptor, because you see here. Okay. Now, once you do that, all these different combinations, now need to do is need to go through exam, essentially. So for this one, you got two things. Positive
Your life in medical school will be like
something like that lah. Think of it like that, so no problem. Very easy to think lah. Okay, so you got positive selection and negative selection. So here, critical, process whereby T cells recognize self-peptides, MHC complexes with low avidity are stimulated to survive. What does it mean? Low. They weak binding lah. Why? You see. And this positive selection involves this thymic epithelial cells. So think of this as your examiner, the thymic epithelial cells that expresses both MHC class 1 and MHC class 2. Can you name me the other cells that actually expresses both MHC class 1 and MHC class 2?
Antigen presenting cells.
Yep. And antigen presenting cells is your dendritic cells, B cells, and macrophage. However, with one exception, you have this thing called thymic epithelial cell. So what they do? They also give. They actually express both MHC class 1 and MHC class 2. So what they do? They perform positive and negative selection. Okay, so they screen for reactivity to self-MHC. So recognition of self-MHC is mandatory. You need to at least pass, okay, the exam, because failure to pass means GG by apoptosis. And there's actually another term called clonal deletion. So at the same time, these double positive cells will actually need to, be certain. Okay, which one do they need to specialize? Either as a CD8 or CD4. You need to choose one pathway. Okay, I know in med school, I mean in IMU or anything, can you do both med school and dentistry at the same time? Do you think it's possible?
No, doctor.
I mean, in term cost-wise, also not feasible, right? But just dealing with med school is already headache already. Imagine you do need to do another part, which is dentistry. So obviously cannot. So they need to choose. Okay, which one? And this actually depends on this MHC class 1 and MHC class 2. So for MHC class 1, what happens? They become CD8. If MHC class 2, this actually allows specialization to CD4 T cells. So positive selection is indicated here in the red dots. Low affinity. They need to actually at least pass, okay? Because here, as you can see, no binding to cell MHC. What's the point? You can't even pass basic. So what happens? Die by apoptosis. Then how about these two? Well, these two is actually will be covered in this one, negative selection. So process in which T cells whose T cell receptors bind with high avidity, strong binding to cell MHC complexes undergo apoptosis. They are killed. What is that? Well, too strong. Weapons. Autoimmunity. Hypersensitivity can happen. So a process to induce immunological tolerance where T cells with T cell receptors react strongly against self-antigens are subjected to apoptosis. Indicated by the blue box here. High affinity, high affinity. Failure, death. Okay, So in negative selection, so autoreactive T lymphocytes will be removed. Okay. However, there is one alternative pathway for T cells that fail negative selection. Why is that? Oh no, they actually failed the negative selection. Yes, it's this one. They are converted to Tregs, T regulatory cells. Now, this one very interesting is that they actually come from the same cohort as the T helper cells, just that they function as a spoiler. They suppress immune responses. T helper cells, they cause immune response. Here, these are actually your jealous T cells that actually fail their exam. So what they do, they go around disrupting the immune response. But they're still very important. Okay, so basically, they are the result of negative selection. Okay, so in summary, for now, try to ignore, I probably ignore this one. Okay, I actually focus more on this. So, positive selection and negative selection. Positive selection is more concerned with the basic, having the actual basic MHC molecule, T cell receptor that can bind to MHC, and as well as a single expression of CD4 or CD8. And negative selection is more concerned with T cell reactivity against self. So as you can see in here, I actually got another one here. Okay, just think of it as in an exam. Okay, so an immature T cell in the thymus, they undergo gene arrangement or VDJ recombination. And during exam, only cells between here, below 80 and above 20, survive. The rest killed. For positive selection, they are killed. Negative selection, they are killed. But some of them, actually, some survive to become Tregs. And these Tregs will do their work in peripheral tolerance. So hopefully with this, you roughly know the idea behind, positive and negative selection. However, not all autoreactive T lymphocytes are removed in the thymus or converted to Tregs. You have some that manage to escape. What will happen if this autoreactive T cells do in the peripheral? So that's why we have peripheral tolerance. Okay, and peripheral tolerance is a bit different. Just now is talking about immature developing T cells. Here is mature T cells. Recognize self-antigens in periphery tissues are rendered incapable of subsequently responding to these antigens. Okay, difference is mature lymphocytes. The mechanism is actually illustrated a bit in the previous lecture. Okay, but we're going through a bit first. So we cover a few of the general mechanisms in peripheral tolerance. You have antigen sequestration, clonal deletion, clonal energy, and Tregs. So first we start with antigen sequestration. So under antigen sequestration, also known as hidden antigen, so what happens here is that our body anatomically, we have some of these, some of our self-antigens are hidden away from the immune system, okay, to isolate self-antigen from the immune system through anatomical positions. Okay, ever heard of a blood-brain barrier? Ever heard of that?
Yes, doctor.
How about blood-retinal barrier?
Yes.
Blood-testis barrier? No, but anyhow, so what does this blood-testis barrier mean? So basically, it means that the blood is actually kept away from that portion. So as you can see, in the eyes and testes, so basically lens, proteins, and sperm, they are actually self-antigens, but they are kept away from the immune system through these immunological privileged sites. I'm sure you heard of immunoprivileged sites before, right? No? Well, that's why the name blood-brain barrier, this barrier implies, okay? It separates the immune system. However, the problem cannot be the main mechanism. Why? Well, only applicable to few antigens, few self-antigens. Okay, we're going to talk a bit on blood-testis barrier. Okay, example. So as you can see, this blood-testis barrier prevents the immune system, or it prevents interaction with, the blood cells in any way or another. Okay, because why? Once you got contact with the sperm, with the immune system, they can cause immune response. So they are sequestered away via the blood-testis barrier. So if this one day is disrupted and you got some white blood cells going in, well, wave your sperm goodbye. Okay, your sperm is going bye-bye in that sense. So that's why you have all this, blood-testis barrier and so forth. Now, with that aside, so that's just an example of antigen sequestration. Now we're going to go into detail with clonal deletion. So same thing, clonal deletion, apoptosis. Okay. However, unlike central torrents, clonal deletion is a minor mechanism in peripheral torrents. Okay. However, not much is still known on this one, on the exact mechanism. People think it's something to do with the fast ligand. But no, we'll see. But still, it's up in the air, okay? Clonal energy, on the other hand, is most common. Now, how does this lead to clonal energy? Still remember your two-signal model? This is where it comes in again. So exposure of naive T helper cell to an antigen in the absence of co-symmetry signals is rendered incapable of responding to the antigen. So self-reactive cell does not die and remains unresponsive. So again, we revisit this, the two-signal model. That's why I keep mentioning the two-signal model. So why? Not only, activation, important activation of T cell, making it to go either Th1 or Th2. Two-signal model is also very important in peripheral torrents. In the sense that, as you can see here, host cells, many cells express MHC1 but do not express co-symmetry molecules. So this means not that many cells can present antigens to T cells, but not all cells can activate naive T cells. You still need that co-symmetry molecule. So if signal 1 takes place without signal 2, no activation will happen, and the T cell will be inactivated or energized. So energized is the antonym to energy, En. Energy means power. Energized is no power. So hence remain indifferent to antigen. And the term here is clonal energy. So this is the major one in the major mechanism in peripheral torrents. Well, self-explanatory. No signal 1 or 2, no activation of T cells. And for that to happen, for T cells to be activated, needs signal 1 and signal 2. Signal 1, T cell receptor and MHC class 2 for T helper cells. Signal 2, depending on the APCs. Okay, here is macrophage or dendritic cells, B7 versus CD28. B cells, CD154 to CD40. Nothing happens. Okay, in signal 2. Even though signal 1 can still bind, without signal 2, nothing happens. How about Tregs? Okay, you see now is how Tregs actually disrupt the immune response. So what they do, even though, well, historically they are known as T suppressor cells. However, it's only in about 20 years ago they start, saying change the name to Tregs. And here what they do is they suppress immune responses. So basically, the balance between Th and Treg cells determine if an immune response takes place or not. And how they do that? Well, we'll come in the next few slides. So here is actually the origin of Tregs. So by now you know that during native selection, what happens? During native selection, either they die or become Tregs. And how you differentiate Tregs from, their T helper counterparts is through this FOXP3. So FOXP3 is a transcription factor. And how they generally do it, generally, is through inhibition of T cell activation, as well as inhibition of T effector function. Now, next slide is actually quite interesting. Okay, how they do that? They produce inhibitory cytokines, IL-10, TGF-beta. Or they even actually directly kill effector T cells by producing granzymes or perforins. Or they actually can do this, metabolic disruption. Still remember your CD25? So this is actually your IL-2 receptor. So T regulatory cells has a high affinity for IL-2. And what does IL-2 do? Still remember? Clonal expansion. They're responsible for clonal expansion. So by taking away IL-2, no clonal expansion happens. And what happens? Death due to cytokine deprivation. And you got adenosine and CMP as well. They can actually disrupt. This one takes away the adenosine. So these are just some more mechanisms. So they can actually even target dendritic cells directly. How? Still remember your... So you got co-stimulators, right? But do you know, you actually got another type of receptors known as co-inhibitors. So these are just extra information. Just extra. For example, this one, CTLA4. They actually bind to CD80 and CD86 of the dendritic cells, which then actually inhibits the dendritic cells from presenting. So, all in all, they inhibit T cell activation, or they directly inhibit the effector function. So that's how it works. Ideal one, well, ideal is a bit tricky. This actually what they do is they deplete tryptophan, amino acid that is actually important for the effector function of T cells. So this is just some of them. Okay. So we covered T cells. Now we are now covering B cells. So okay, so for B cells, they are quite similar, just that happens in the bone marrow. And the major mechanism is this one, clonal deletion. You also have clonal energy. And here, quite interesting is this one. You have high affinity and high avidity, but the outcome is the same. It's either clonal deletion and this one, receptor editing. For now, high affinity and high avidity, they are actually different. But most importantly is, okay, they can recognize self-antigens with high affinity and high avidity, may result mostly in clonal deletion. However, you still got chance. Unlike T cells is once you fail, you die. B cells, they are actually much more flexible in the sense that they can get to reseed their paper. And how they reseed is to do this, called receptor editing. So as you can see, self-reactive B cells with high avidity or high affinity result in clonal deletion. However, they can still get to reseed their paper. They get receptor editing to generate a non-self-reactive cell. So they got second chance in that sense. However, if they repeat too much, ultimately also become this. Okay, so it's not indefinite. For low affinity, on the hand, is this one, clonal energy. So that's what you need to know. Okay, low avidity, low affinity is energy. And for peripheral tolerance for B cells, quite similar. Mostly is clonal energy. Because same thing, signal 1, signal 2. And you also have deletion. Then you also got suppression. So this is actually another special case, suppression by inhibitory receptors. So the main one for this one is this one, energy for B cells. And this is the inhibitory receptor, CD22. And how does CD22 do? As you can see, it inhibits the B cell receptor. And that's about it. Okay, so what happens if you lose immunological tolerance?
So the body will produce an immune response to your self-antigens.
So you have autoreactive lymphocytes and autoreactive antibodies. This will lead to autoimmunity. So yeah, that's about it. Okay, so I think we got time. Why don't we do a bit of exercise? Okay. Name the cell. You tell me, yeah? Okay, CD3.
T cells.
Sorry?
T cell.
T cells, good. Specific T cells or all T cells?
All T cells.
Good. How about CD4?
Helper T cells.
Good.
Cytotoxic T cells.
Good. Wait, huh? Okay, here, if I say T-Rex, is it still okay?
Yes.
Good. So how do you differentiate these two, huh?
T regulatory cells have the FOXP3.
Yep. So one way, remember this, huh? Okay.
The dendritic cells of the macrophages.
Okay, good.
B cell.
Good.
Is it the T regulatory cell?
Remember this one? B cell. Okay, I'm going to open up another section. Now you're going to tell me is what is a function? Wait, how to insert? Okay. Into the So what does CD4 do actually? Sorry. CD3 do first.
Do they differentiate to the effector cells?
So you remember they because the T cell receptor is too short, so they need something to transmit. So the antenna for the T cell receptor is very short, so they need something to transmit into the nucleus. So that row pumps down to the CD3. So they transmit signals to the nucleus intracellularly. How about CD4?
Co-receptor.
Receptors, but specifically, what do they do?
It provides stability.
Stabilize binding between TCR and MHC. Okay. How about cytotoxic T cells? CD8.
They kill they direct killing the foreign antigens or your
Okay, so I think the point of this is I'd just like to know is what is the function of these cell surface molecules. I know the function of CD8 T cells they do is that they directly kill viral-infected cells, tumor cells, cancer cells, and so forth. But so what is the role of CD8 molecule?
Is it the same as CD4?
Yep. But again, CD4 binds to what? 2 or 3? 1 or 2? MHC class 1 or MHC class 2? Which one?
Class 2.
CD8?
Class 1.
Yep. How about FOXP3?
It's a transcription factor.
Yep. So by now, just focus on being a transcription factor. That's about it. B7?
Is it a co-stimulatory signal?
Yep. Co-stimulatory molecule. And what does it do?
It produces signal too.
Same thing for this one, right? For CD40. Okay, how about CD22? Well, it's this one, okay? Aha, okay. Might just go a bit more. Okay, can I add more?
Doctor, could you
Yes.
explain a little bit more about the CD22 inhibitory receptor?
Oh, okay. So what is CD22? So CD22 is an inhibitory receptor on the B cell. So it's actually what happens here is if it binds to self antigens, this actually will cause inhibition of the B cell receptor. And B cell receptor also can, it prevents the activation of the B cell, as shown here. So this is actually just some details, okay? But overall, just remember CD22 is just an inhibitory receptor on the B cell that actually regulates, B cells. So it's just like a disruptor, just in case, B cells get activated for no reason. So that's what CD22 does.
Oh, okay, thank you.
Okay, so I'm sure by now you know what, antibodies, you can find them everywhere, but I'm actually going to ask you is this one. What is the function of IgM?
Is it neutralization as well as it acts as a BCR?
Yep. Oh, BCR, neutralization. What else? Activation of the complement system. Okay. How about IgE?
Neutralization as well.
Generally, all antibodies, they neutralize. So very easy answer. However, for there are actually some specifics, okay. So for for IgE, what do they do specifically?
ADCC, degranulation of mast cell and basophils.
Because of allergy, in allergy. So how they do that is because IgE, once you need to get sensitization first, then this IgE will bind to the mast cells, to the IgE receptor, SC receptor. Yeah, it's a bit more, you need to actually go into a pathogenesis of allergy in order to get this in detail. Okay, but here, IgE is mostly this. How about IgG?
The opsonization.
They are the only type of antibodies that can do that. They can actually cross from the placenta into the fetus. Okay, and in terms of affinity, or in terms of affinity, they're actually the strongest. Have you encountered the terms avidity and affinity? Apart from my lecture, of course.
Actually, doctor, what's the difference between the two?
Okay. I was worried about that actually, because all this time I always have questions on that one. Okay. Wait, I probably show you. I need to show you. I'm actually going to go to internet and look. Wait, where is it? Wait. Mhm. Okay, so as you can see here, can you see the screen?
Yes, doctor.
So this is actually the difference in the different isotypes of antibiotics. You got 5 of them. So IgG, IgM. So IgM is considered to have high avidity. IgG is high affinity. So difference is how do I put it? So avidity is the sum of all interactions or sum of all binding. So as you can see, IgM has how many binding sites? 10, right? So as a result, the sum of all binding in combined is actually known as avidity. Whereas IgG has high affinity, means the individual binding between IgG and the antibody and the antigen is very strong. So that's actually the difference between these two. So normally IgG is known as having high affinity. IgM is high avidity. So that's actually the difference between this. Okay, I'm going to look at questions. Oh no, okay, never mind. That's not a question. That's a statement. Anyhow, any questions so far? Again, questions. If I can't answer your question now, don't worry. Please direct your questions to your student rep and ask your student rep to send to me in a document, in a Word. Preferably by today. I'll try to answer everything by tonight. Then I'll send it back. Or during tomorrow's Q&A session, if you got even more, feel free to ask. Okay, so we've got 2 chances. Just that for tomorrow's session, I'm just worried that tomorrow I'm scheduled for vaccination. They haven't tell me what time yet, so which is a bit annoying. So just to inform you first lah. Then if I can't attend, I'll just I say I apologize and probably arrange another Q&A session on our own, maybe in the weekend, I suppose. Is that okay?
Sure, doctor.
Okay. So with that,
Sir, can
yes,
I ask a question?
Yes, please.
So the more binding sites there are, then the lower the affinity? Or is it not necessarily like that?
Not necessary. But the idea, I mean, how you visualize avidity versus affinity is the binding of between just one. That's affinity. Whereas avidity is a sum of all binding sites between a molecule. Yes, between molecules.
Okay.
So
okay.
at this point, I mean, just roughly get that idea first. Later on, if you encounter this again, you see. Because at this point, just get used to these terms. You probably will encounter these terms many times actually.
Okay.
If you proceed, if you do further reading, you encounter this. So
Okay,
it's just a way to introduce you to these terms for now.
Okay.
Again,
thank you so
everything
much.
here is just introduction. If you want to go even deeper, you can actually go and look into the references and all that. But again, the idea here is the idea of the lecture is actually to introduce you, just roughly the basics. You'll see in the references it gets even more complicated.
Okay, thank you so much, Doctor.
So get used to this one first, the basics first. Later on, then when you do your further reading, it makes more sense. Okay, so with that aside, if I'm not mistaken, this actually I actually going to send to you this lecture slides within this chat. Wait, I'm going to send to you now. You can actually download directly. Where is it? Because I did some changes, so these are actually the much more recent ones. So I sent to this chat is better lah. I'll actually update this in the e-learn also again. So that's why you see the V2 there, version 2. So receive. So you can actually download that directly and use that. Okay, with that aside, again, questions, please ask your student rep to contact me directly. If you got any questions, send to them and have them sent to me. Or if you want one-to-one interaction, use Teams to contact me, direct message. Then I'll actually respond accordingly. So that's another way. All right. So with that session, I'll conclude the session. All right. So all the best and see you.